In this video, we're going to look at histone acetylation. And if you like this content, check out the links in my bio to the self-paced course or the MCAT Mastery program. Both of those give you access to over 500 pages of the highest yield MCAT content just like this. So let's zoom in on this image here. Right here is a chromosome, and what this is showing is all of these blue circles are wrapping up into a highly condensed structure to form that chromosome. Each one of those blue dots is actually this entire structure right here. That structure is made up of 8 of those proteins, and those are called histone proteins. The DNA is this red strand here, and it wraps around the histone proteins, and then we have this purple clamp thing that basically holds the DNA to it. Histone proteins have a lysine, and let's scroll down to see what that looks like. The lysine has a positive charge, and the backbone of DNA is a negative charge. And when that DNA, which remember is the red strand, when that is wrapping around the histones, the interaction between the negative charge on the DNA and the positive charge on the lysine on these histones is what helps to hold the DNA to the histone proteins. And I do not have the lysine side chain drawn on the green dots in this image, so just imagine that it's there interacting with the red string of DNA. When the DNA is tightly wrapped around the histones, it's not really accessible to the machinery that can transcribe it. So the genes in the DNA right here would not be being transcribed much because they're held too tightly to the histones. And that's where histone acetylation comes in. Over here we can see that an acetyl group has been added to the nitrogen on that lysine side chain, and that gets rid of that positive charge. And now the DNA backbone is not as attracted to the histone proteins. This image shows how the histones in the DNA unwind, and that makes these sections of DNA accessible to the machinery for things like transcription. So now the genes on this section are expressed at a higher level because they're available for that transcription machinery.